Hi all, today we're gonna talk about a simplified version of the immune system. This process is very important because without it, we would be infected by all the viruses in the environment. So first of all, when a virus tries to get inside of the body, the first barrier that it meets is the skin. So as you can see, the skin acts like a shield, so it can keep the virus away from us. This is why we must enjoy this beautiful gift that the nature gives to us. Moreover, to protect ourselves from those bodies who finally get inside of our body by the mouth, the nose, or the eyes, we've got a second line of defense composed by cells that can neutralize the viruses. There are many kinds of those cells, such as macrophage and neutrophil. Once again, this defense line is totally inborn. And also it's very essential for us to get this line of defense to survive. Now, Patricia will explain to you how the contamination is made by the virus when it enters in our body.
Okay, so now the viruses are in the body. So when they are in the body, they won't go infect all of the cells. It's only the specific cells. To explain that, there's proteins on the cells and on the viruses. So when the viruses start the cells to contain the proteins that fit with the proteins contained on the viruses, they come out. So if there was proteins on the viruses like that, and on the cells like that, that won't fit, so it won't infect those cells. But when they saw proteins that fit, that means that it's the right kind of cells. So those proteins just locked, and the DNA here contains in the cells goes into the infected cells. Once it's there, there's a copy was made of the DNA, because the DNA is too precious to be used. So the copy is called the RNA. So when the RNA is produced, it's read by the ribosomes of the cells. So how it works is that ribosomes goes on the RNA like that, and comes and brings new amino acids. So it produces all of the new proteins required to produce new viruses. Well, when all of the compounds are made, they all coalesce, so they all come together. So all of the compounds you have here, DNA, and go together, so the new viruses are produced. So when they're all produced, there's a new mechanisms that make the cells break. So it's called the lysis of the cells. So the cells there's a holes made, and all of the viruses get out of the infected cells, and the infected cells dies. And now all of the new viruses can go into the organisms and kind of contaminate all new cells out.
The last part of the response of the immune system to the virus is the apoptosis and the antibodies reaction. And first, we have an antigen-presenting cell with its major histocompatibility complex. This is a protein where the antigens of the virus are presented to an T helper cell. A T helper cell is a lymphocyte. A lymphocyte is a cell that is a part of the acquired part of the immune system. These cells have a specific use against a specific virus. So the cell secretes cytokines. So the T helper cell, the cytokines of the T helper cell go to the T killer cell. This cell is the one that do the apoptosis. With is CDI, the complex, and is another protein. It go to an infected cell, and when he is near this infected cell, he secretes perforins. The perforins are the proteins that do the apoptosis. Apoptosis is the reaction where a cell have a hole in his body, so the cell die. So, as you can see, this is proteins. The water get off the cell, so the cell lose all its structure and is dead.